Good morning, good evening, good afternoon, wherever you happen to be in your time zone, and thank you for attending this PGC meeting today. I'm going to tell you about three major collections in Finland that utilizes some of the Finnish characteristics and special characters in the Finnish population. So they are FinnGen, the Super Finland project, and the Northern Finnish Intellectual Disability Cohort. So the success of genetic studies in disease genetics, specifically, are lying on basically four cornerstones: the population isolation, the nationwide health registers, the long-standing epidemiological studies which have further developed to biobanks, and then of course knowledge of genome data. First, diving into what the national registries in the Scandinavian countries are. They are ways to record the usage of the healthcare services. And these are very similar in all Scandinavian countries, not just in Finland. It means that wherever in the country you visit a hospital, your diagnosis is recorded in a central register. The same thing when you purchase a prescription drug. These registers were originally developed for administrative purposes, but since they accumulate a lot of data of healthcare service usage, they become very interesting resources for research. And although they are silos as such, the typical individual identification number or social security number, whichever way you like to look at it, then combines this data together once you get the appropriate permits. The second aspect, the population isolation. The history of Finland, or the current population history of Finland, stems a few thousand years back, where a small number of settlers were mainly living on the coastal regions of Finland. But then in the 16th century, the Swedish King Gustav Vasa demanded Finns to move also to the eastern and northern parts of the country, which resulted in a second very strong internal migration and multiple bottlenecks. And what happened then in all Nordic countries after the 18th century is a rapid population growth, which obviously then resulted in all the typical characteristics of a population isolation. The nationwide registers that I was describing, what is great about them is that every single individual is, every citizen, every resident is recorded, and they stem back decades back in a digital format. Some of them go back all the way to the 50s, but I think the most important time point is the late 1960s, when the hospital discharge data, in the cause of death data, and the reimbursement data came into usage. And this obviously provides you an opportunity to look at longitudinal data, because this health data is available from birth to death. And this is one of the very key characteristics of this data, even if it doesn't include some symptom level data. It's ICD codes, ATC codes, but the great thing is its longitudinality. And this is what the FinnGen research project is based on. It's a project that was initiated in 2017, and with the idea that it utilizes genetic strategies to understand disease mechanisms. It's aiming to collect 500,000 individuals, which is roughly the 10% of the population, genotype them, and impute them against a national or population-specific deep whole genome sequence backbone, and then integrating the National Health Register data, which results in a data set of 500,000 individuals with both genome and health data for our association analysis. This project is a public-private collaboration, a research project where all Finnish biobanks, which exist in all university hospitals around the country, all universities are involved with a medical school are involved, as well as the Institute of Health and Welfare and the blood transfusion center. Also, the Finnish Biobank Cooperative is a partner there, and currently 13 pharma partners are together with us working on our scientific goals. We are currently, this is a 10-year project, and we are now just completed year 4, and the sample collection will go on for still a little bit less than a couple of years, and so at which point we are full with our collection, and then really have an opportunity to further focus on the analysis. Where are we in respect of the collection? We have currently collected 472,000 participants, which means that we are slightly ahead of schedule and seem well set to reach our goal. This includes not quite yet 200,000, but it will include some 200,000 legacy collections and then 300,000 prospective collections. And the prospective collection, what is crucial here, that most of them come from hospital biobanks, typically university hospitals, which means that they are specifically enriched for diseases treated in these places. So if you compare to UK Biobank, this is a big difference in the content of the sample. We produce data sets every 6 months. The current data freeze age will consist of 350,000 individuals, and there are more than 4,000 endpoints. And all of these FIVAS analyses done from these data freezes are made public after 12 months from there, once they become ready. And if you look at the type of phenotypes that we have, then the special thing is again that the age is relatively high here, which mean age, which means that there are quite a lot of healthcare events already. The mean number of health events per individuals is 340, including some 186 drug purchases. This means that there's quite a lot of data. And how we build then these phenotype endpoints is that we combine data from different registers, whether drug usage or hospital visits, and create a strengthened endpoint by this in this way. And this needs quite a lot of understanding of how the data works, or the national register data, how it functions, what are the strengths, what are the drawbacks, and so forth. And how do we then access the data? We have 2 levels. Both of them work in the Google Cloud space. One is where we have the summary level data, the results, in other words, from GWAS and FIVAS analysis. And then there is the secure environment And this is something to stress in the way that those familiar with the European data regulations, which have become quite stringent. So this is coping with all those requirements and still being able, we are able to access the data from outside European Union. And the idea is that it's a secure environment where you cannot take data out. You can analyze it, you can look at it, but you cannot copy it out from there or download it to your own computer. What has been characteristics in analyzing these data is that there are a large number of Finnish-specific variants that even in very extensively studied, genetically very extensively studied diseases like type 2 diabetes. All the ones with stars here marked are Finnish enriched alleles and Finnish specific hits. And this goes through, this is a characteristic through all traits that we actually have studied, that there are typically Finnish enriched variants. The other way you can look at it, since we have the medication data from all individuals and the entire country, so you can ask questions the other way around. That what about those ones who use medicines? Do we see variants enriched that actually give additional information for us, not just from the basic diagnosis? And this seemed to be the case, at least in some of the cardiovascular statin medications and those related medication GWAS. So when we look at the numbers of mental health diagnoses in FinnGen, out of the 321,000 individuals that were in data freeze 7, so any mental health ICD codes was observed in 76,000 individuals, but even more individuals had depression medication, some almost 8,000 more. And depression diagnosis was then seen in 33,000 individuals. Schizophrenia in 11,000, schizophrenia, schizotypical and delusional disorders, 11,600, schizophrenia 6,000, and bipolar almost 6,000 individuals as well. A place where you can look into the distribution of diagnoses and how they have been constructed from the register data is called Risteus. And that is under, if you go to the FinnGen website, you can be guided to this Risteus website, which is open for everyone. And you can see various the age distribution of the individuals, years of first diagnosis and comorbidities. Quite a helpful site to look to get an understanding how our endpoints are constructed. Then we move to the second collection, which is the Super Finland collection, and this has been critically supported by the Stanley Center. A collection that was done over years 2016 to 2018. It consists of a little bit more than 10,000 patients with psychotic disorders. All samples have been GWASed on and exome sequenced, a little bit less than 900 also whole genome sequenced. Almost 90% of the individuals have consented for IPS research and can be recontacted. And as of now, a little bit more than 5,200 PBMC lines have been collected for iPS cell generation. And this map is showing the distribution from where the patients are, which means that they pretty well represent the population density also in the country. There, as you can imagine, there's a huge number of hardworking people behind it. They don't all fit here in the slide, but they worked very hard over the years to first to collect them and now later to analyze them. The diagnostic groups in Super are so that a little bit more than half of them, a little bit less than 6,000 have a schizophrenia diagnosis, 1,600 a bipolar disorder, and the next largest being the schizoaffective disorders, psychotic depression, and other psychosis patients. From the recruited individuals, we can clearly see that as expected, the education completion is lower than in the general population. They are less married than non-psychotic individuals in the populations and so forth. So we have an understanding that quite a lot of these people are chronically ill and represent that type of psychosis spectrum. We also have collected questionnaire, cognitive test data at the time of collection. So we don't have longitudinal cognitive tests, just at the recruitment. We have the medical care register data and also the blood plasma stored. The third collection and the last one is the Northern Finnish intellectual disability cohort. We currently have a little bit more than 3,000 participants, and they are all collected from the northern regions. And if you remember, when I was describing the late settlement movement, which happens after this 16th century, these were areas that were very sparsely populated in small villages. And we can see from the frequency that the prevalence of both intellectual disability and schizophrenia are higher in these areas. And that was one of the reasons that stimulated us to collect individuals that don't have a clear ID diagnosis from this area. Currently, most of these have already been exome sequenced and GWASed, and for instance, just for interest, 56 schema gene variants are observed in 80 carriers. So it's kind of interesting that since we are these have a majority of mild ID cases, which is slightly unusual for these type of cohorts. It's interesting to compare now the variant carriers in specifically this group and then in the super psychosis study, and indeed same variants exist as well as same genes, but having different variants in these two collections. And I thank everyone for listening and hope that these cohorts provide an opportunity for good harvest. Thank you.